So before we begin, we want to consolidate the relevance of this project to the learning outcomes of the course. Primarily, the video inspects the role of telomeres in DNA replication and cellular reproduction. So, we'll review the basic steps and key enzymes involved in DNA replication, covering LO2 and LO3. We also reinforce that carriage of DNA is a key characteristic of life, covering LO4. There'll be heaps on nucleic acids, supported by case study examples as contextualization to the discussed role of telomeres, which henceforth further covers LO4 and LO5. Therefore, the examination of these learning outcomes aligns significantly with Theme 2, which unsurprisingly is cell biology and cell architecture within the BABS1201 course. Our case study allows for greater understanding of Theme 2's third dot point to describe important cell structures and relate these to function, thus building the background knowledge for the understanding of the further themes 3 and 4. So, let me give you a short revision of lecture information. Alright, we already know that DNA is a double helix made of those repeating nucleic acids, hydrogen bonded to each other. And we also know that they sit in the nuclei or nucleides of eukaryotic and prokaryotic cells respectively. You should already know that they're bunched into chromatids and make up the chromosomes, and if you paid attention to the first lectures, you would know that some DNA is circular, like in bacteria, and some is linear, like in vertebrate eukaryotes.
The Grim Reaper's alarm clock in your body is based on the telomeres getting too short. See, telomeres are made of repeating TTA-GG nucleotides at the ends of linear chromosomes, as pictured here. When telomeres get shorter, one can say that the DNA in that chromosome is older and nearing the end of its reproductive life. So when DNA is unable to replicate further, cells stop dividing. If they did, cancers occur. A literal countdown to death. Remember that each DNA replication cycle actually loses some DNA length because of limitations in the DNA polymerase. The protein, remember, needs a 3' end to attach nucleic acids on and cannot replace an RNA primer unless there is a 3' end already available for them to add stuff onto. At the end of linear chromosomes, there is no such helping support, and the polymerase just kind of gives up and says scuff it. In subsequent DNA replications, a certain length of DNA is lost from the ends of the chromosomes, and the chromosomes get progressively shorter as replication cycles keep repeating. To stop important DNA information from being lost, telomeres sacrifice themselves in place of the important stuff. They're like a little builder's hard hat at the end of chromosomes, protecting the actually important DNA repeats from losing information. These hard hats, they can get scraped and damaged quite a bit with use, but they get the job done.
But telomeres don't simply just shorten naturally. There are several other things, artificial influences, such as oxidative stress, that can shorten telomeres. The triple G sequence in the telomere is pretty weakly bonded, so it can be readily damaged by oxidative stress. These high-intensity stresses cause the DNA double strand to break even without DNA replication. This is normally accompanied by other genomic damages. For example, you get cancer. A single strand break in this picture is possible and will screw up the DNA replication process. So either it stops the helicase for both strands, or the broken strand loses length. So everything after, which is the telomere bit as well, is lost. And telomere shortening rates are directly related to the frequency of these single strand breaks. So there's a potential theory. Cells which have been exposed to high levels of oxidative stress have a higher probability of senescence and thus mutating. Unfortunately, a concrete understanding of the physiological causes behind aging is still undiscovered. However, it's been accepted for a while that telomere shortening has a significant association with increasing likelihoods of contracting age-related diseases such as cancer, organ failure, and Alzheimer's disease. So, to that extent, it's possible to equate telomere shortening with aging itself. So, we can answer, what causes aging? As, what causes telomeres to shorten more quickly?
Oxidative stress is caused from the exterior environment and poor personal lifestyle choices, having an impact on the telomere length. Firstly, we need to define what oxidative stress refers to. There are two important cells which generate in our body, free radicals and antioxidants. Free radicals are any serious reactive molecules containing an oxygen atom and uneven numbers of electrons. Antioxidants are things that give an extra electron to free radicals, to stop them from being so angry. When an imbalance between these two exists, that's oxidative stress. Excessive amounts of free radical damage the DNA in your body, which happens if your cells don't generate enough antioxidants. Things that causes this is smoking, drinking alcohol, not exercising, being overexposed to sunlight, and not getting enough sleep. Environmental factors in the biological sector is very broadly categorized. It includes inflammation, physiological stress, and exposure to carcinogens, all of which lead to an accelerated telomere length reduction. Australian summer is a classic example of death. Experimental findings on lifestyle influences on telomere lengths are often contradictory to each other. While one study on lifestyle influence on telomere lengths showed no association between telomere lengths and exercise, another study did show an indication. One possible explanation is that these experiments use leukocyte telomeres, which may remain relatively unaffected by these lifestyle influences compared to other cells throughout the body that may be inspected in other studies.